Hello, so as requested, I'm going to do a short video about antibodies. So lesson objectives then are to define the terms antigen and antibody. We'll look at the structure of antibodies and think about how the structure relates to the function of antibodies. We'll outline the mode of action of antibodies. We'll talk about different types, so opsonins, agglutinins and antitoxins. And then we'll look at the primary and secondary immune response. So thinking about an antigen, first of all, it's a molecule that stimulates an immune response. So it's a recognition molecule. Often they're glycoproteins that you get on the surface of cells in the body. They've got a very specific shape, and they act a little bit like a barcode. So our body can detect foreign antigens, and then that will generate an immune response. And that immune response that we looked at in the last few lessons then means that the B lymphocytes then will start to produce antibodies as a result. Those antibodies then have got antigen binding sites which are complementary to those foreign antigens. So B lymphocytes then produce these antibodies as a response to this infection. So B lymphocytes then, they'll recognise these foreign antigens, they'll release antibodies, and antibodies can attach the antigens because they have this complementary shape, and it will make those antigens or the pathogens harmless, and they can do that in a number of different ways. So we're going to look now at the structure of antibodies. They've got this nice Y shape that you can see here, but we're going to zoom in and look at the structure in a little bit more detail. So the antibody then is made up of 4 polypeptide chains. So we've got 1, 2 of these heavy polypeptide chains and 2 of these light polypeptide chains. Now each of those polypeptide chains has got what we call a constant region. That's this bit here. And the constant region is the same for each of the antibodies. Now that's quite useful because it means that they only need to have one set of instructions for each of those polypeptide chains for this constant region. So the base sequence is going to be the same for each of these constant regions there. So the only bit that needs to change in terms of the DNA code to make these polypeptide chains is the little bit that's going to code for these bits at the end, which are called the variable regions just here. So those are the antigen binding sites, and those need to be specific to each antigen. So each pathogen's got different antigens, and each of those then will have a specific antigen binding site. And all of these polypeptide chains, these 4 polypeptide chains, then held together by disulfide bonds. So these are incredibly strong bonds, which is why we have them in the antibodies. And it creates also this hinge region here, so a bit like a door hinge. It allows this antibody to be flexible so that it can bind to multiple pathogens at the same time. So we're going to think now about how these antibodies work. So we've got a few different types. So we've got antitoxins. In a nutshell, those antitoxins will neutralise the toxins that are produced by particular pathogens. We've got agglutinins that we'll look at in a little bit more detail. In a nutshell, they'll clump the pathogens, clump those cells together so that the macrophages can engulf them. And we've got opsonins here as well. And again, in a nutshell, those will help to increase or encourage process of phagocytosis. So with the agglutinins then, they work by clumping together the pathogens and then delivering the pathogens like a giant Scooby snack to the macrophages, and they can all be engulfed together. And so those will travel around and they'll bind to pathogens there, and then they'll deliver those pathogens to the macrophage, and the macrophage can engulf all of those pathogens at the same time. So it just speeds up that process there. With the opsonins then, the opsonins have got two binding sites. So they've got the antigen binding site that we mentioned at the beginning of the lesson when we looked at the structure, but they've also got a binding site that is complementary to receptor sites on the macrophages. So it will bind to the pathogen with the antigen binding sites, and then it will bind to the macrophage, and again, like a delivery service, they'll deliver the pathogens to the macrophages, and the macrophage then will engulf that pathogen and digest it. They'll also digest the opsonins as well, so those antibodies, the opsonin antibodies, will also get digested at the same time. So we're going to think now about the primary and secondary response. So the primary response is the first response that we get when we first encounter any kind of infection. So we've got our first infection here, and you can see that it takes a bit of time. Here it's taken 5 days for antibodies to be produced, and that's because it takes time for that process of clonal selection to take place when they select the correct type of BLT lymphocyte. Once they've selected the correct type of BLT lymphocyte and we've had the clone expansion process, so they've divided by mitosis and then differentiated, and the B plasma cells then will start to produce antibodies. But we don't get that many antibodies being produced in the first time we encounter an infection. But after that first infection, then we'll create memory cells. So B memory cells in particular are really important in this process. And that means that if we get the infection again, very quickly those B memory cells can differentiate into B plasma cells and start to produce antibodies. So we're getting antibodies being produced straight away. They can also divide by mitosis to make more copies and then make even more antibodies, which is why we've got a much, much higher concentration of antibodies with the secondary response. And if we look at, we can do a little bit of a comparison here. So in the primary response here, you can see we only get around about 30 arbitrary units with the antibodies that are produced, whereas with the secondary response, you can see around about 145 arbitrary units, so much higher concentration, and those antibodies remain in the blood for much, much longer. Good, yeah, so in a nutshell then, we've got the foreign antigens first being detected, takes time then for that process of clonal selection, and then the antibodies start to be produced, and doesn't really rise high enough to destroy the infection. It takes a bit of time to destroy that infection. With the primary response then, because it's taking a bit longer to fight off that infection, to start the production of antibodies, a person would start to feel symptoms here. With the secondary response then, much, much quicker. You've got a much higher concentration of antibodies and a much quicker response. So with the secondary response here, when you've got those memory cells, you won't actually, it's so quick that you won't feel those symptoms. So it's incredibly quick with the secondary response. So I would suggest in your notes then that you describe and explain the graph. So have a look at it. Don't forget that point evidence explanation that we've talked about before. So say what you see, back it up with some data, and then explain, why does it take 5 days to start to produce those antibodies in the primary response? Why does it take just 1 day? Okay, any questions? I'm on Team App in our normal lesson times, so don't hesitate to contact me via Team App. Thank you. Bye.